Alright, this is going to be a quick overview of the Osmosis Lab for Bio 1441. This is a lab that has several steps to it because there's a long incubation period, so we have to keep them busy. So first of all, this is the mass balance table, the dilution table for the students to help them get set up. I put this on there because I have students do bulk dilution, so each table does a dilution. They do 300 mils of 0 to 1 molar sucrose, and they're going to need to make 300 mils of each one, and then each table is going to draw 50 mils from each of those concentrations for the experiment. Students are also going to core 7 potato discs and cut them into 5mm long pieces, just like they did with the beets. Each one of those is going to go into one of those concentrations, and one will go into an unknown concentration. But before they do that, they have to mass each piece individually and record that mass. Just to warn you, some students will want to record the potatoes mass all at once, which is pretty much useless, so make sure they record each one individually. Once they've all done that, which will take longer than you expect, but about 45 minutes in or so, you should be able to drop your potato pieces into the various concentrations of sucrose, and they should then work on their hypothesis. And make sure you tell them that as they think about what their hypothesis will be, that sucrose does not cross the cell membrane easily, but water can. They're then going to work on, we're going to not do the lab report workshop, we can delete that bullet, but we're going to work on the Elodia observation, which will be putting Elodia in a hypertonic and a hypotonic environment and using microscopes to observe what happens to the cells. And we will also do a endocytosis observation, which is not listed on this slide currently. So you'll want to explain how osmosis works. The way I like to do it is to bounce around the room kind of randomly, explaining that molecules move as long as there's energy in the system, and that as molecules cross membranes, like water crossing a membrane, I'll stagger out into the hallway and come back again, saying that water can move in and out of a cell. But if I moved out into the hallway and sat on top of an elephant, like water crossing a membrane and getting stuck to a large sucrose molecule, it would not cross back into the cell. It would be stuck. And that's what we can see in this slide here, explaining why the water levels change over time in this U-shaped tube. You can then explain how plant and animal cells vary in their responses to these various environments. In a hypotonic environment, plants actually thrive. Their vacuole, it will fill the cell, like 90% of the volume of the cell, making this plant cell very turgid, like a water balloon inflated in a shoebox, so it has some rigidity. That's how a stick of celery, for example, is crispy, while an animal cell would lyse in these conditions. In isotonic conditions, water moves in and out of the cell at an equal rate, which is good for an animal cell, but not so good for a plant. That vacuole will kind of shrink a little bit, and the cell will become flaccid, so think of a floppy piece of celery. In a hypertonic environment, an animal cell will shrivel and a plant cell will plasmolyze. It's basically dehydrated and the plasma membrane pulls away from the cell wall, and pretty much the cell's toast at that point. I have a video that you can show them of an egg in various conditions. I also have an exercise you can do with them just predicting what would happen in these different situations. Then once they get everything set up and we've talked about osmosis, and then what I do is I have them get the microscopes out, or we may have them out already for you, 2 per table, and talk to them about basic microscope safety, about how to focus and not to ram their objective lens into the slides, things like that. We talk about the major parts of the scope and their functionality, and then students will make a wet mount. You'll probably have to show them how. One half of the table will put Elodia just in water, which is the normal condition, and they'll see the cell walls, these big cells full of little chloroplasts. If they're lucky, the chloroplasts might even be moving due to cytoplasmic streaming. I have a video of that up here if you want to show them. And then the side of the table that puts it in salt water will end up with cells that are plasmolyzed. This is a grainy image, but you can see the cells here are plasmolyzed. The cytoplasm has formed a gooey ball in the middle of the cell. It's pretty much dehydrated. So they'll be able to see that. I asked them to take pictures of this with their phones and show me what they've observed. After that, they can take a drop of Tetrahymena culture, hopefully it'll still be alive next week, and a drop of India ink, and the Tetrahymena will actually bring the ink into their cells. Presumably that ink sticks to bacteria, or perhaps they're eating the ink itself, and we can see them in their food vacuoles inside the cell. So we can kind of see endocytosis occurring. And you can tell them that that's just an observation, so they can actually see this thing they learn about in class. They might be able to see the cilia twitching on the Tetrahymena, and you can tell them that they'll be doing a follow-up experiment using Tetrahymena and endocytosis when they take cell biology. Lastly, they're going to collect their data, put it all into a class data collection table, and then answer a few questions, including what solution would be isotonic to the potato and what concentration was the unknown. And so you have to explain what a standard curve is to the students, and then I show them an example, and a lot of times our unknown is like 3 molar or 5 molar. Here's an example where it calculates out as 0.21 molar, just giving them an example. But I wanted to show them an example of the data because they have a hard time interpreting it. They fail to realize that if the change in mass of the potato, which is what we're recording, change in mass, if the change in mass is above 0, that means the potato gained mass. And if it's below 0, it lost mass. So if their hypothesis was the potato would lose mass as you increase concentration, technically there should be a little more nuance there for that hypothesis to be correct. The change in mass will decrease, I guess you could say, but both of these concentrations in this case actually gained mass rather than lost mass. So just think about that as you're showing them the data. The homework that I give them then will be a mini, or what should I say, it's a lab summary, a digital lab summary. I have them write a hypothesis. I have them write a rationale for the hypothesis, put a figure in that Word file, and also a 2 or 3 sentence conclusion. So the hypothesis is worth a point, the figure is worth probably 15, and then the conclusion is worth, let's see, where am I at? The 16, probably about 4 points, so a total of 20 points is what I am assigning for that assignment. And I can send you more info on that if you want. Anyway, that is the lab for next week. Let me know if you have any questions, and have a great week. I'll see you later.